Hi, everyone. I'm Alan Cantor from Boston Children's Hospital, and I want to thank Amanda and the RRP for the opportunity to present this clinical trial to you. We're very excited about this. So this is the work of a lot of people, but Leah Cunningham from the National Cancer Institute and I will be representing. And Leah's really done a lot of the work in really developing the protocol. So I'm going to begin by just giving you some of the rationale for this study, some of the background, some of the data. And I'm going to hand it over to Leah to really go through the protocol and really talk about some of the practical aspects of it. Okay, so as I think everyone knows here that RUNX1 FPD is due to a mutation in one of the two RUNX1 gene copies. So everyone gets 2 copies of most genes, one from your mother, one from your father. But importantly, in this disorder, there's still one wild type copy or one normal copy left here. And so we and other groups have asked the question, can we find means to enhance the activity of this residual normal protein to try to compensate for the deficiency from the other one? And many proteins don't kind of always work at their fullest capacity. And so there may be ways we can try to get it to work at its full capacity to do this. So early on, with support from the RRP, we developed a method to measure RUNX1 activity in a way that we could test thousands of drugs kind of at the same time using some robotic platforms at a screening facility at Harvard Medical School. And RUNX1 is a transcription factor. So that's a protein that works by turning on and turning off other genes in the genome. And for this particular assay, we studied one of a gene that RUNX1 normally regulates called c-MPL that's very important for making megakaryocytes and platelets. And what was very striking when we did this is, and we again screened about over 1,000 drugs, that quite a few of the drugs that were scoring as hits all were inhibiting this very same enzyme or pathway. And this shown on the bottom here are just 3 of the drugs I'm showing here. What I'm showing here is the relative amount of RUNX1 activity with increasing concentrations of these different drugs. And you'll see in blue is our assay that's really looking at RUNX1 activity, and in green is just a control for any effect of the drug that is not working through RUNX1, anything independent. And I think hopefully you can see for all these drugs that we're getting a dose-dependent increase in the activity, all giving about a 50% boost in RUNX1 activity, shown here, and really not much activity with the controls. This seems to be working through RUNX1. And this really fit with some earlier work from my laboratory and some other groups that have been studying RUNX1 regulation. So this is a cartoon of the RUNX1 protein. And towards the end of the molecule is something called an inhibitory domain. It's part of the molecule that normally dampens or restrains its activity. And what we've shown is that this part of the molecule can be chemically modified by something called a phosphorylation or phosphate groups. And these are put on by these same enzymes that we had found these inhibitors were active. So these enzymes called Sarc-Abl family kinases add this phosphate group to this region, and that makes RUNX1 less active. But we also knew this was very, very dynamic and that there were other enzymes that could remove this phosphate and make it more active. And so what these inhibitors can do is they can kind of reduce the amount of phosphorylation happening in the first place and shift the balance towards a more active form of RUNX1. So this is the idea behind this. And so really the next step was really to test these drugs in various what we call preclinical models. So these are cell lines or animal study models that can tell us that there is really activity here and that we should proceed to human clinical trials. So we and our colleagues have done this in a number of ways. The first is using some what we call induced pluripotent stem cells. These are cell lines that are derived from patients, and the one I'm going to show you is from a patient that has a deletion of one of the 2 RUNX alleles. And these are cells that can develop into anything in culture in the petri dish, and we can culture them into mature megakaryocytes, which is obviously an important phenotype to study. And this is just a graph showing the fold increase in the mature megakaryocytes. And on the left in blue is just sort of the control. And then all the way on the right in yellow is cells that we've actually replaced the defective rungs copy with a normal copy. So we've genetically rescued them. So this is about as best as we could expect to obtain. And you can see with the imatinib and the nilotinib, we were getting some rescue activity. And Paul Liu and Anupriya Agarwal, also through the RRP and as part of this NIH natural study, have also examined this using RUNX1 FPD patient bone marrow cells and can culture them in vitro into megakaryocytes. And what they found also is if you treat these bone marrow cells with increasing concentrations of imatinib, here going from right to left, they also observed an increase in this mature megakaryocytes. So that was promising. And then Lucio Castilla, another RRP investigator, has generated a nice mouse model of RUNX1 FPD, and he's been studying in part the DNA damage response. And so what he does is he takes bone marrow cells from these mice, takes them out and puts them in culture, and he gives them a low dose of radiation and then scores DNA damage using this test that where these green dots represent areas where there's been DNA damage, and you can quantify this. And what he found is with the RUNX1 FPD mice compared to the wild type controls is that they had an exaggerated response to the DNA damage. There were more cells with more of these foci compared to the wild type. But importantly, when he took these bone marrow cells and first treated them with imatinib and then did the radiation, he could almost completely rescue this and fix this problem. And we've seen something very similar using some human cell lines that we've generated, that we've engineered to be lacking one of the RUNX1 copies. And here we're using something called doxorubicin as another way to induce the DNA damage. And we're measuring DNA damage in a different assay, something called the COMET assay. And here we're plotting what's called the tail moment, which you can just think of as the amount of DNA damage or DNA damage burden. And if you just look at the two red bars, you can see that with RUNX1 deficiency, there's a very exaggerated response, as we kind of knew from prior work. But importantly, if you pre-treat the cells with imatinib, you could almost completely abrogate this. So this is all very encouraging, and has really led us to really start moving this in towards phase 1 clinical trials. And we had a number of drugs to choose from, but we ended up choosing imatinib, also known as Gleevec, for this, for a number of reasons. So first of all, imatinib's been around for a long time. It was first FDA approved in 2001. In fact, it made the front cover of Time magazine then. This was really the first example of precision medicine. It was very exciting at the time. It's currently FDA approved for a number of leukemias and other disorders or similar related disorders. There's pretty low toxicity, and Leo will probably talk to you more about this. And again, over a 20-year experience using this drugs, and some of these patients take it for years at a time. We knew that the bloodstream concentrations of imatinib in patients taking a standard dose, which is 400 milligrams by mouth daily, corresponded to the levels that were showing improved RUNX1 activity in our experiments. And also, the Gleevec patent expired in 2016, so it's now generic, and this has some, practical implications for cost and availability. So with all of this, we are moving ahead with a phase 1b study of imatinib to increase RUNX1 activity in participants with germline RUNX1 deficiency. And this trial now is up on the clinicaltrials.gov. Here's the number. And we expect to be able to start enrolling patients as early as probably this week or next week. So now I'm going to stop sharing, and I think Leah is going to start sharing her screen to tell you more.
All right. Well, thank you so very much. For anyone joining late, my name is Leah. I'm at NIH. Really appreciate the invitation. This is my absolute favorite meeting of the year, every single year. It's a huge honor, pleasure, and privilege to be here. And I'll go ahead and jump into the details of the imatinib study. I couldn't be more grateful for the foundation provided this morning by Nancy, by Katrine, by David, and everyone else who spoke so far. So I can very quickly go through some of the background and hopefully make up some time. So this is the schema of the study. We are going to start with screening here of everybody to make sure that they are generally at their baseline health. There are two groups from there, RUNX1 unaffected or healthy controls who are donated blood or marrow, and those who are affected by RUNX1, who will also be split into two groups. The top group here is imatinib dose escalation, meaning that we are going to start with a very low dose of imatinib and then increase those doses. And then the second group from there is imatinib dose expansion, so that the dose that we identify in arm 1 will be given to arm 2, but for a longer period of time to make sure that things are safe and tolerated. From there, folks will be followed up for 30 days, and then the study will be done. This is a more granular look at those details. So starting here at the purple star, we are going to start at the arm 1 dose escalation. We are starting at 25% or 1/4 of the approved dose of imatinib. And we are starting basically, each dot here represents a person. Blue means no dose limiting toxicity, and red would mean a dose limiting toxicity. So for example, we're starting at 100 milligrams for 3 participants. If there are no dose limiting toxicities after 28 days, then the next group gets enrolled and starts at 200 milligrams, also 3 people. From there, again, we look at everybody for 1 month, make sure there are no dose limiting toxicities, and then we start at 300 milligrams for 3 patients. As an example here, though, I put in, okay, well, what happens if one person has a dose limiting toxicity? Under those circumstances, we would enroll another 3 patients and then see what we see and see if this is tolerated or not. If it's still well tolerated, there's no dose limiting toxicity, then we have another month of observation, and then we end up at 400 milligrams, which is the final dose level that we're planning to evaluate, because that is the approved dose for folks who would be treated for CML, for example. So therefore, under these circumstances in this example, 400 milligrams would be the maximum tolerated dose, and we would give that dose, whatever it ends up being, to the expansion cohort for 12 patients for 12 weeks, or for 3 months, basically. So most imatinib adverse events are grade 1. They're mild or asymptomatic, for example, and anything that qualifies as a dose-limiting toxicity on the previous trial would be anything that is more than grade 2. So as Dr. Young pointed out in the previous talk, we know that folks are coming in with changes from the general population in their blood systems, their blood counts, their platelet counts rather, their platelet function, and of course, gastrointestinal. And I'm personally probably the most worried about gastrointestinal symptoms such as nausea. So we are giving everybody Zofran, for example, that they can take with them in case they experience nausea. So the goal here is to minimize side effects as much as possible. So the objectives of the study are safety and to identify the dose for the dose escalation phase, and safety pops up here again. So this is both for the escalation phase as well as for the expansion phase. And the most important part of the study is to figure out before and after imatinib, are there changes in the bleeding assessment tool? Are there changes in platelet aggregation, if we can measure that, as well as in the electron microscopy, looking at the platelet ultrastructure, and as well as the pharmacokinetics. So as Alan pointed out, folks with RUNX1 certainly have had imatinib along the way, just not in a prospective organized trial like this. Anybody who had ever had a RUNX1 mutation, who had AML or MDS or any other indication, would have had that done, but not in an organized fashion. And that's the purpose of the study. So there are a ton of exploratory objectives here. I'm sorry that this slide is busy. Alan went over already the target gene expression and the DNA damage response. And for anyone who attended the meetings last week, Larry went over the very sensitive flow-based assay that he's using to look at platelet function. And we want to know also, is dose related to any of the secondary endpoints, 300, 400, what is most biologically meaningful? So any one of these things has the potential to become an endpoint or an aim on the follow-up phase 2 study. I do also want to point out that Coleman Lindsley has very kindly agreed to work with us to look at variant allele frequency from an exploratory standpoint under 5%, for example, and his level of sensitivity is is very low. He also spoke at the meeting last year. So in terms of inclusion, we are starting with adults from a safety perspective. People will have adequate organ and marrow function. They will not be pregnant. And for every affected person, of course, they'll need a confirmed pathogenic or likely pathogenic mutation, a history of bleeding, such as an abnormal bleeding assessment tool score, history of needing Amacor platelet transfusions, and also abnormal aggregation and platelet electron microscopy. They will be at their baseline bone marrow with normal, for them, flow cytometry, normal cytogenetics, absence of deleterious secondary mutations on the TSO 500, and the ability to swallow imatinib pills. This is kind of a reiteration of, or the opposite of the inclusion criteria, so that nobody would have a history of a problematic secondary mutation. Platelets would be above 65,000, no history of blood cancer, allergic reaction to imatinib, the need to take medications that would change imatinib plasma levels, and that would include things over the counter like St. John's wort, for example, that would significantly change imatinib levels, any uncontrolled illness or heart conditions. So this is the calendar, and so a history and physical, standard labs, platelet studies, and the marrows are optional. You know, we hear you, not everyone wants to give a marrow. If people are able to give them, you know, with fentanyl or Versed or, you know, whatever they need, then that's appreciated but not mandatory. So day one is a busy day at NIH. There are many pharmacokinetic blood draws that will be done with a blood drawing IV, so it's really only one stick. Days 7, 14, and 21 are telehealth by home, including a LabCorp lab slip so that people don't have to charge their insurance or copays, et cetera. And then we hope that people will come back at day 28 for follow-up, because that will help us a lot with the interpretation of the data. Now, because the RUNX1 patient population is among the most dedicated and impressive and organized groups of patients I've ever had the privilege of working with, I know that although this looks like asking a lot to do a bleeding assessment tool every 7 days from home to do these patient-reported outcomes measures, to me, this is probably one of the most important parts of the trial, because this is going to tell us exactly how patients are feeling. And these are hugely important in FDA approvals of medications, you know, for example. So although labor intensive, very important, can be done on paper pencil or on the computer. So for arm 2, for the dose expansion, this is 84 days. It'll go longer, the exact same idea in terms of getting a baseline on day 1, doing a whole bunch of telehealth every 7 days to assess how people are feeling, getting their basic labs at home. And then the last day of arm 2 is day 84. And then this is probably the most, you know, labor intensive to fill in a questionnaire every 7 days. So hopefully that is not too much to ask. So in terms of frequently addressed questions, I can kind of fly through this because David just went through this. Well, a lot of it anyway, in the sense that there's no direct benefit to participants of safety studies, but many people report to us that they do feel that their participation may help others. We do hope that participation will help us identify meaningful phase 2 endpoints. Insurance and copays are not required. Clinical care consults and close monitoring are. NIH does assist with travel, lodging, and meals, and most of it is telehealth from home. So hopefully that increases the convenience. Most people have been, you know, to NIH, but for anyone who hasn't, this is the clinical center here. Just out of view is the Safra Lodge for adult lodging. My personal opinion is the Children's Inn is the crown jewel of the entire campus, and a lot of patients and families have told us that they don't mind staying there. And a lot of the pediatric enrollments can go up to age 30 to stay at the Children's Inn. And then here are the NIH study contacts. Ashley and Rebecca are the research nurses. Yi Lu is the genetics counselor at NCI on this study. And I'm the principal investigator of the study at NIH. So very quickly through future directions, as we talked about in the last session, if you have a big problem, you want a big toolbox. And we as a community, you know, have kind of are filling this in and will be filling this in. So this study will open this month. This should complete in 1 year. And at that point, we would know if imatinib is safe and well tolerated. We would know the recommended phase 2 dose and the most biologically relevant endpoints. There is a stem cell transplant trial that is also cooking that I'll just quickly throw in. I threw in a slide for that. And then we've talked also through the meeting last week and also this morning, there are many other things under consideration, many other RUNC studies that include etanercept, sirolimus, and harvesting, for example. So what would 2025 look like under those circumstances? There's a lot of question marks here. But by then, would we have enough data to open the imatinib efficacy study? And what would that look like? Would it be a single arm? Would it be a comparison against one of these items, depending on how those look? Would it be a combination of items? And historically in oncology, things have always worked best by combination due to a lot of escape mechanisms and others that I'm not going to get into at the moment. We talked about risk score. I showed this at ASH in 2019, and we're still trying to figure this out. Depending on what your mutation is, whether or not you have somatic mutations, are they good for you or bad for you? Do you have cytogenetic abnormalities, FISH abnormalities? About 2% of RUNX1 patients have normal cytogenetics, but abnormal FISH. So a first marrow is always important to look. Is there frank dysplasia? So when we looked through all of the data in the natural history study, many patients, their transplant note said, came with AML, and that might have been true. But if you went back 5-6 months of the medical records at other hospitals before that, a lot of those cases actually arose from MDS. And so I'm just going to throw that in there because of the discussion of medical records, from last week's scientific session, how critically important it is to get as many medical records as you can, and to get ahead of the kind of cut paste that sometimes affects medical records, and how some groups see more AML than MDS AML, for example. And also, of course, the stroma is abnormal. So even post-transplant, folks with germline RUNX1 deficiency who have been transplanted still have hypocellularity. Why is that? Is it a problem of both? So, since then, we're still considering, what is the best diagnostic and monitoring model, as we talked about in the last session. We have to think about conditioning toxicity, genotype-phenotype correlation. We still don't know, but we do know that catching things early is still absolutely the best policy if you're going to need treatment. So just a very quick, slide on the transplant protocol that I'm just going to throw in here, that we are trying to minimize chemotherapy exposure as much as possible. So this is kind of standard transplant fare up here for very high-risk people who already have a lot of disease. But for those who might only have CCUS or what's called low-grade MDS right now, can we get away with just an antibody, which is against CD117, along with very low doses of TBI or fludarabine? And better yet, the next generation of this protocol, can we get rid of this altogether and just give 2 antibodies, one's against the 117 and one's against 45, and then you're trying to deal with a low-risk chemo-free protocol. So that's kind of the goal after this one. And I got approval for kind of expedited review of this one. So this should open next year as well. So it goes without saying that this is just an incredible community, that none of this would be possible without the patients' families, referring providers, the RUNX1 research program. So all of the collaborators and all the work that you have seen here, both this week and last week. Paul and the entire group at the National History Study, NHGRI. Sung Young, I've been working now for the last year and a half, and she has given me a lot of protected time to finish these protocols, which has been great. Steve Pavletic is kind of the deputy of I appointed him the deputy of the imatinib trial at NIH. He's probably best known for 20 years of great work in GVHD consensus trials. Many other people, including people in Sung Young's lab, who have helped to move things along. The myeloid malignancies program, who's been very supportive, and a lot of the RUNX1 lessons that I have applied to this program to move things along there. We are now the poster child for the NCI Division of Cancer Prevention. We're the first hematologic malignancy trial in this group. So all of the fellows that rotate through there and all of the senior leadership is very excited. And I'm also thrilled that I got permission to be in the North American Australian Alliance, whatever the new name is going to be, as an official duty capacity to help with the next generation of consensus. Everybody in the Department of Laboratory Medicine, and most importantly, all of the people who have provided consults in hematology, GI, dermatology, in pulmonary, allergy, immunology, neurology, gynecology, all of these people who have seen so many RUNX1 patients at NIH and are very well versed, who can provide consults for these protocols. So thank you so very much, and I'm happy to take any questions along with Alan.